Okay, good morning, class, and welcome to the discussion of Chapter 2, okay, Part 1. Okay, so this is the Part 1. We will be having the Part 2. I will also be uploading the Part 2 or the video file, okay, following this video. Okay, so for Chapter 2, we will be talking about the cell structure and taxonomy or the classification and the naming of the different microorganisms. How we're going to name or how are we going to write the scientific name or the binomial nomenclature. Okay, so let's start with the chapter outline. Okay, you have your chapter outline. In this chapter, you will be able to learn about, okay, number 1, prokaryotic cell and its function. Eukaryotic cell structure and its function also. And number 3, differences between your prokaryotic and eukaryotic cells. Number 4, microbial classification and nomenclature. Now, all in all, in chapter 2, the topic will revolve around just differentiating, okay, your prokaryotic cells from your eukaryotic cells. Now I know I've already introduced these terms in the first chapter, right? Okay, so in chapter 2 we will be going deeper, okay, and understanding these types of cells and the different microorganisms or organisms belonging to these types of cells, okay? Yeah, so let's go to the introduction, okay? The first topic will be prokaryotic cell and prokaryotic cell structure and its function, okay? Now, on the last chapter, diba, we've been talking about the different types of organisms that you will be studying in microbiology. Now, let's just have a quick explanation of what types of cells composes your plants, animals, bacteria, or archaea, your fungi, protozoa, algae, and your virus. Okay, let's go with first your plants and animals. Now again, you will need to take note of this because what I'm going to tell you that is not included in your course pack. Okay, so for your first organisms, you have the plants and animals. Now, when you say plants and animals, these are organisms composed of eukaryotic cells. Okay, so they are entirely composed of eukaryotic cell, meaning they are not unicellular. When it comes to prokaryotes, you have two types of organisms. Okay, prokaryotes, meaning unicellular. Isang cell. They are composed of only one cell. So you have two types of organisms under your prokaryotes. You have the bacteria and the archaea. Okay, now bacteria and archaea are the only two organisms under prokaryotes, but they are not they are not under one classification. Why? Because they have large or huge difference. Okay, magkaiba ang bacteria sa archaea, although they are the same when it comes to classification of the number of cells or the prokaryotes. So that is number 2. Now, you also have the third one, which is composed of the fungi, the protozoa, and the algae. Okay, now these three class are also classified as eukaryotes. Okay, kaya nga natin pinag-aaralan dito sa microbiology, diba? We've included the eukaryotes because there are also microorganisms like your fungi, protozoa, and algae that is composed of many cells. Okay? So, wait lang class. I will just check if I'm still recording this. Okay, we're good. Okay, this is still recording. Okay, so that is number 3. You have the fungi, protozoa, and algae as eukaryotes also. And then you have the number 4, which is the virus. Now, the virus, as what we've tackled during the first chapter, again, this is not classified whether it is a eukaryote or a prokaryote. Why? Because your viruses are non-cellular. It is not composed of cell. Why? It does not fit in any organizational scheme. Hindi siya, it does not belong to prokaryotes. It's also not belonging to your eukaryotes. Why? Because its genetic particles replicate, okay? The viruses are able to replicate its own genetic particles, but they are unable to perform the usual chemical activities of living cells. Okay, so they can replicate their DNA. Kaya nilang ipasa ang DNA nila sa iba, but when it comes to chemical activities such as, for example, producing their food, breaking down their food, producing certain types of proteins, enzymes, hindi nila kayang gawin yun. Okay? So, they are not considered as a living cell. Okay? So, those are the 4 groups that will be tackled, okay, in the chapter 2 classification. Okay, so let's go to the first topic, prokaryotic cell structure and function. So we will be talking about the different parts of your prokaryotic cell. So when you say prokaryotic cell, again, ito sila yung bacteria. Okay? Most of the examples that I will be showing in here will be talking about the bacterial cell. Okay, so your bacteria were the earliest and simplest types of living organisms to inhabit the earth. Okay? Bacteria, it is the bacterial ancestors were the first living cells, they are the first living cells to appear on earth. Okay, sila yung pinakaunang living cells that were recorded to live on earth. Okay, so pinaka-ancient ancestors ng lahat ng organisms actually in this world is your bacterial cell. Okay, very resilient. Now, all bacteria are called prokaryotes, okay, coming from the Greek word protos meaning primitive and karyon meaning nucleus. Okay. So, your prokaryotes, by the way, class, yung ginagamit dito in your course pack, the spelling that it's using is letter C. But you can also use letter K, okay, for prokaryotes and eukaryotes. Pwede rin K, pwedeng C, it's just the same, okay? Okay, your prokaryotes are morphologically much simpler than eukaryotes. So, tapos na tayo in explaining that. Let's move on. Okay, so these are the chief distinguishing characteristics of your prokaryotes. Most of this class I've already discussed during the first chapter. Okay, so what are those? Number 1, they do not contain a true nucleus because their DNA is not enclosed within a membrane and is located in a discrete region in the cytoplasm called nucleoid. Okay, so diba? As I've said, the DNA of your prokaryotes are not enclosed, okay, with the membrane. So, kapag hindi siya naka-enclose sa loob ng isang nuclear membrane, that means that it is not a true nucleus because your true nucleus is bounded by a membrane. Now, since it's just spread in a certain location or in discrete region in the cytoplasm of a bacterial cell, you will call it as a nucleoid and not a nucleus. Okay? So, nucleoid siya kapag hindi nakabalot or not bounded by a membrane. Okay, next. We have they do not contain internal membrane-bound organelles. Unlike your eukaryotes na napakaraming membrane-bound organelles, okay, its interior appears morphologically simple, or it has few internal structures. Now, this let's just consider the fact that the bacterial cell is so small. So syempre, it will not contain a lot of organelles. Kasi it's the structure of your bacterial cells or the prokaryotic cells are just very simple. Okay? So, okay, next. We have number 3, the DNA is not associated with histone proteins. Okay, when you say histone proteins, again, these are chromosomal proteins found in your eukaryotes. So the function of your histone proteins is to bind the strand of DNA material inside the nucleus. Okay, so when you have histone proteins, the strand of the DNA will be coiling, okay, to form condensed or dense forms of chromosomes in order to fit inside the nucleus. So, yun yung pangatlo, histone proteins. Number 4, almost always bounded by a chemically complex cell wall. Okay, so meron siyang cell wall just like your plants, diba? The difference between your animal cell and your plant cell is that your plant cell contains or is bounded by a cell wall, diba? A rigid type of protecting wall, okay? While the animal cells, they do not contain cell wall. Now, the same with plant cells, your prokaryotic cells also contain a chemically complex cell wall, okay? And their cell wall almost always contain the complex polysaccharide peptidoglycan. So, yan, bacterial cells always contain your peptidoglycan. So when you say polysaccharide, what does it mean? It means that it is composed of what? Many units of sugar, okay? Polysaccharide, this is a topic in your biochemistry. So I know meron na kayong background dyan. Okay, let's go to number 5. They usually divide by binary fission during this process. Okay, the DNA is copied and the cell splits into 2. Okay, so we've already discussed this. Your prokaryotic cells does not undergo the mitosis and meiosis division. Okay, so it only multiplies using the binary fission. So I have an example here of the binary fission, of how your binary fission occurs. Okay, now as you can see here, this used to be a whole single bacterial cell. Okay, but when you look closely, it's starting to separate with each other. Okay, there is division in the middle of the cell. Now this is what you call binary fission. Okay, ganyan yung appearance ng isang bacterial cell undergoing binary fission. Now, these are the steps, okay? You have here your E. coli or Escherichia coli cell. So it contains a cell wall, it also has the plasma membrane, it contains its bacterial chromosome or the nucleoid, and it also has the origin of the replication of the DNA. Now, when it undergoes binary fission, what happens first is the chromosome replication begin. So, it replicates its DNA material. Okay? Soon after, one copy of the origin moves rapidly toward the other end of the cell by a mechanism not yet fully understood. Now, there's no much explanation on how this itong origin ng replication ng DNA class, di ba? Isa lang dito sa original cell, iisa lang siya. Now, pagdating dito sa first step, the origin of the replication of DNA becomes 2. Okay, magre-replicate siya. And it will go to the different or the opposite regions of your bacterial cell. Okay, so in the step 2, dalawa na ang origin niya. One on the left side and one on the right side. Okay, two replication sites. Again, ano yung nire-replicate dito sa origin? The DNA material. Okay, so after that, replication continues. One copy of the origin is now at each end of the cell. So, meanwhile, the cell elongates. Now, when another copy of an origin, okay, is transferred, okay, to the right side. So, syempre ngayon, dalawa na yung nagre-replicate ng DNA material. Now, in order for this DNA or for these bacterial chromosomes to fit, okay, inside the cell will elongate. Okay? Kasi mas marami na yung na-produce na bacterial chromosome. Now, on the third step, replication finishes. The plasma membrane grows inward and a new cell wall is produced. So after the replication class, you will have two bodies of bacterial chromosome. So dalawa na yung DNA material. Okay, the first one is coming from the left origin and the second one is coming from the right origin. Now, when this happens, when the replication is accomplished or finished, there will be a division, okay? An inward growth that is happening at the center of your elongated bacterial cell. And then later on, separation will occur. Okay, so mapuputol siya in the middle and the final products are two daughter bacterial cells. Okay, so this is the process of your binary fission. This happens usually in bacteria class or in prokaryotes. Okay, next. So in your course pack, merong included na images of the structure, okay, generalized structure of a prokaryote. Now the sample given here is a sample of a bacterial cell. We have the different parts of the cell, okay, placed in here. First one, you have the pilus. Okay, pilus ang tawag sa kanya class kapag iisa lang. Now, more than one, ang tawag is pili, okay? Plural form ang pili. You also have the capsule in here. Okay, now your capsule, ang definition niya sa inyong course pack. Okay, this is a jelly-like outer coating of many prokaryotes. So the capsule can be either jelly-like or solid. Okay. Next, you have the cytoplasm. Okay, your cytoplasm. So meron ding cytoplasm ang inyong prokaryotic cells. You also have the ribosomes. Okay, so what is the function of your ribosomes? So, ang ribosomes class when you compare it with the eukaryotic cell, diba? So, eukaryotic cell, you will only find ribosomes on certain parts of the eukaryotic cell. While in prokaryotes, you will find ribosomes spread, okay, sa lahat ng parts ng cytoplasm, okay? So, it is spread all over the cytoplasm. So its function for production of proteins. Next, you have the cell wall. Okay, and then you also have the plasma membrane, and then you have the nucleoid, and then you also have the plasmid. Okay, what is a plasmid? Okay, your plasmid class is very unique. These are just found in prokaryotic cells, and its shape is, it's like a circular double-stranded DNA. Bilog siya, and it contains two strands of DNA molecules. Now, this is characterized as a extrachromosomal DNA molecule. From the word extra, okay? Extra siya. Hindi siya kasali. Separated siya sa nucleoid containing your DNA. Okay? your plasmid is a DNA molecule that is separated from your nucleoid. Now, one unique characteristic of your plasmid is that it can also replicate, and it replicates independently. Okay? When you say independently, hindi siya nagre-replicate kasabay ng nucleoid. Okay? It is not, the replication of your plasmid does not depend on your nucleoid. It can replicate independently because it is separated from the nucleoid. Okay? So the plasmid is actually unique to your prokaryotic cells. Now, you also have your flagella or flagellum for singular. Now, usually the flagella class is just used for locomotion. Then you have your capsule. Ayan, ito yung wider view niya. So, your prokaryotic cells, especially your bacteria class, has 3 coverings. Okay? So, when you say prokaryotic cells, meron sila, their unique characterization is that they contain 3 coverings. One is the outer covering is your capsule. Okay, the second covering, the middle part, is the cell wall, and the third part is the plasma membrane. So, nakita nyo sa balot pa lang, sa coverings pa lang ng bacterial cell, it is very, or your prokaryotic cells, it is very safe. Okay? It is covered by 3 layers, and yun yung pinakamahirap na i-penetrate. Okay? Kaya hindi siya mabilis na mamatay. Now, compared to your animal cells, ilan ang covering ng animal cell natin? Or your eukaryotic animal cells? Only 1. What is that? It only contains plasma membrane. So, plasma membrane, walang cell wall. And compared to the plant cell naman, eukaryotic plant cell, your plant cell contains 2 coverings, the cell wall and the plasma membrane. So, take note of that, the difference between the coverings of your prokaryotic animal cell and plant cell. Okay, what's next? So, we're done with this. Okay, so this class, I gave you another material or image that contains the different definitions or functions of each part of your prokaryotic cell. So, dito nyo na lang tingnan, meron din ditong kanilang definition and function. So, you can use this as a study material. You just read that. Next. Okay, let's go to the structures external to the cell wall. So, these structures class are found beyond, okay, outside your cell wall. So, if you look at here, so kung ito, the middle covering is your cell wall, outside this are the external material. So, this includes your capsule, your flagella, okay, your pili, and your so on. So, let's just take them. The number 1, you have glycocalyx, second flagella, third eggshell filaments, and fourth your pili. Okay? So again, your glycocalyx class, this is the capsule. Okay? Ito na siya. The general term for the capsule, or sometimes slime outside or protecting your prokaryotic cell, you call that as glycocalyx. So may 2 types siya. Pwedeng solid, which is the capsule, and pwedeng slimy or smoother version, which is the slime layer. Okay, so this is what they look. Okay, this is your eggshell filament. This is your pili. Okay, so let's move on. Let's go to glycocalyx. Your glycocalyx is a sticky mass that surrounds many prokaryocytes or prokaryotic cells. Okay, when you say prokaryocytes, prokaryotic cells, yan sila. Okay, so sticky mass siya. It is sometimes called as the extracellular polymeric substance, or EPS. Okay, you call that as EPS. Okay, Now, when you say ETS class, this is a natural, they are, meaning your glycocalyx is a natural polymer of high molecular weight secreted by most of the microorganisms found in their environment. Okay? So, natural polymers of high molecular weight, and they usually class your glycocalyx or your extracellular polymeric substances. They establish the functional and structural integrity of biofilms. Okay, meaning your glycocalyx is a material used as a support system to biofilms. When you say biofilm, ito na yung tinatawag nating thin slimy film. Okay, found at the outer surface of the bacteria. Now, when you say biofilm class, usually nakikita nga sila, diba, sa mga prokaryotes natin, especially the bacteria. Now, the function of your biofilm, please take note of this, it adheres to surfaces. Okay? Kaya nga sticky material yung glycocalyx ninyo because the glycocalyx is classified as a biofilm. It adheres, dumidikit siya sa certain surfaces. Okay? So your extracellular polymeric substances are again the one that establishes the structural and functional integrity of biofilms, meaning they are the main component of biofilms, especially your glycocalyx. Okay? So sticky. They need to adhere to surfaces in order to build a colony or a population of their own. So, dumidikit sila. Kaya nga, ang mga bacteria natin is hard to remove on surfaces. Okay? So, your glycocalyx is also a gelatinous polymer composed of polysaccharides or polypeptides or both. So, ito na yung components class. These are the components of your glycocalyx. Number 1, meron siyang polysaccharide. So, meaning, polymer siya ng sugar units, okay? And your carbohydrates. And then next, you have your polypeptide. When you say polypeptide, group or conglomeration of your proteins, okay? Composed of many units of proteins. Or pwede ring, sa glycocalyx natin present ang both your protein and your sugar. Okay? So, it is made inside the cell. So, your glycocalyx, it is produced inside your bacterial cell and is just excreted to the cell surface. So, coming from the inside of the cell, excreted outside the surface of the cell. Next. Okay, wait lang. Okay, next we have number 2. Okay, your glycocalyx is classified either as capsule or slime layer. Now, for the capsule, this glycocalyx is organized and firmly attached to the cell wall. Kapag yung glycocalyx is nakadikit ng mabuti doon sa inyong cell wall, okay? Because the cell wall again is the middle part, diba? So, your cell wall, it is where the glycocalyx is attached. Now, when the glycocalyx is firmly attached and organized outside your cell wall, then the glycocalyx is classified as solid. So, ano siya? Capsule siya. Okay? So, presence of the capsule can be determined by the use of negative staining. Such as, one example ng negative staining natin is your India ink method. The India ink method. So, in an India ink method class, what happens in there is that the background is stained while your organism remains clear. Okay? Sa India ink method natin, ang nangyayari, your organisms are not stained. They remain clear. So, ano yung nasa-stain? Yung background. So, in that you can clearly see the microorganisms such as your bacteria, kasi ang na-sustain is yung background at hindi yung bacteria itself. Okay, so that is the India ink method. So negative stain yan siya, class. Okay? So usually ang India ink method, class, this is used, no, it can be used to determine if a cell has a gelatinous capsule. So, yan, your capsule's outermost structure of the bacteria. So, as you can see here, outermost layer siya. And then, okay, number 2. Okay. This is also important in contributing to bacterial virulence. When you say virulence, class, this is the degree to which a pathogen causes disease. Okay, gaano ka-virulent yung isang bacteria or yung isang pathogen in causing disease to the host. So, ayan. By the way, class, not all your bacteria contains a capsule. So, hindi lahat ng bacteria merong capsule. Okay? Now, one way in making your capsule a contributing factor to bacterial virulence, meaning minsan, kapag present yung capsule, mas virulent yung inyong bacteria. Mas matagal siyang mapapatay. Why? Because your capsule it causes more disease because no phagocytes can engulf them. Okay? So, if merong capsule yung bacteria ninyo, class, hindi siya may engulf ng phagocytes present in your immune system. Okay? Diba? Meron tayong mga phagocytic cells found in your immune system. Examples natin, we have, for example, your neutrophils, your monocytes, diba? Macrophages, dendritic cells, yung killer cells ninyo. So, they perform phagocytosis, wherein they use their plasma membrane to engulf the large particles. Okay? So, endocytosis yan siya. Now, when your bacteria contains capsule, hindi siya nakakain nitong phagocytes natin. Okay? So, in that way, it can actually contribute to the virulence of your bacteria. Kasi nga, hindi siya napapatay when the capsule is present. Okay? Now, you have another type of glycocalyx, the slime layer. Okay? So, it is unorganized and loosely attached to the cell wall. Okay? Unorganized siya. Yan. So, loosely attached. So, hindi siya solid. So, slime layer siya. Yan yung pinagkaiba nila, class. You just take note of that. So, this is the example of the appearance of a slime layer and a capsule layer. Okay? Pag slime layer, ganito siya. Pag capsule solid, napakatigas. Okay? So, next. Let's go to functions of the glycocalyx. Number 1, your glycocalyx may protect the bacteria from phagocytosis, making them more virulent. So, ito na yung sabi ko kanina. They are able to cause more serious disease because they are not easily killed. Okay? So, one example natin, you have examples of bacteria that contain capsule. You have the Streptococcus pneumoniae. Okay? This causes pneumonia when the cells are protected by a polysaccharide capsule. So, kapag yung strain ng Streptococcus pneumonia or Streptococcus pneumoniae contains a capsule, they are the ones that cause your pneumonia disease. Kapag hindi siya coated na capsule, hindi siya nagkakause ng pneumonia. So, ganun yung ibig sabihin niya, class. Okay? So, next one, you have the unencapsulated S. pneumoniae. So, or Streptococcus pneumoniae. So kapag hindi siya okay, they are readily phagocytized, okay, nai-engulf sila phagocytes ninyo, and thus they cannot cause pneumonia. So yun yung example class ng importance ng capsule para sa mga um, microorganisms. When they are not covered with capsule, they are engulfed and they die. Okay? So, hindi na sila makakos ng disease. So, only encapsulated Bacillus anthracis can cause anthrax. Now, when you say anthrax class, this is an example of a serious infection. Okay? So, ano ang nagkakos ng anthrax natin? Your bacillus, your encapsulated, meaning may capsule, covered bacillus anthracis. Ito yung organism. Okay? Now, what happens in your anthrax is your bacillus anthracis kasi class is naturally found in the soil. Nakikita siya saan? Sa lupa. And your bacillus anthracis affects domestic and wild animals. So, originally class, your anthrax, the disease anthrax, originates from animals. And nalilipat lang siya sa human. It is passed through human. Now, what happens in anthrax? You will experience fever, chills, shortness of breath, dizziness, cough, and nausea, and marami pang iba. Pero ito lang yung kinuha ko na sample. So, you will sometimes vomit. Okay? So, your anthrax is a type of disease class that is classified as zoonosis. zoonosis. Zoonosis siya. When you say zoonosis, it is able to transfer the disease from animal to humans. Okay? Pag sinabi mong zoonosis yung isang disease, napapasa siya from animals to humans. And one example of a zoonosis disease is your anthrax. Okay? So, ito yung itsura ng Bacillus anthracis ninyo. Okay? Next, we have the second function. Your glycocalyx may allow the bacteria to attach to various surfaces in order to survive in its natural environment. One example ng natural environment ng mga bacteria ninyo is fast-moving streams, sa rocks, sa plant roots, and also you will find bacteria sa human teeth. So, yung presence ng glycocalyx, it enables your bacteria to remain stick to the environment. For example, is your Streptococcus mutans. Ito yung bacteria na nagko-cause ng teeth decay. Okay, yung pagkabulok ng ngipin. So, if may bulok kayo ng ngipin, meaning merong Streptococcus mutans sa bunganga ninyo. Okay, and you will need to find a certain cleaning product that will kill this bacteria. Pero kung ganito nakabulok yung ngipin class, much better na ipabunot nyo na. Okay, to not further worsen the situation. Okay? So, di ba, yung bulok ng ngipin kapag nandiyan na, ang hirap-hirap ng matanggal and it will further destroy your teeth. Na dahan-dahan na magdedecay yung teeth mo because your Streptococcus mutans contains the glycocalyx. Okay? Next. Third function of your glycocalyx, they may serve as a source of nutrient by breaking it down and utilizing the sugars when energy stores are low. So, di ba kanina nabanggit na one component of your glycocalyx is polysaccharide siya, meaning meron siyang sugar. Now, if kulang yung nutrient source doon sa environment ng isang bacteria, it can break down its own capsule or glycocalyx in order to get the sugars found in the glycocalyx. because it is composed of polysaccharides. Okay? So, yan yung another function niya. Fourth function, this may protect your bacteria against dehydration. Okay, if we look deep inside this single microorganism, we will see that it is made up mostly of water. Okay, ang pinakakalaban class ng mga microorganism is dehydration. Kaya nga class, kapag when there is a wet surface, you will expect that it contains bacteria. One example is your armpit. Okay, pag basa yung armpit ninyo, katagalan bumabaho siya. It produces odor, and the presence of the bad smell represents the presence also of bacteria. Okay? So, 'di ba pag unang basa yung armpit, hindi pa naman siya mabaho, pero katagalan bumabaho siya because tumutubo na yung maraming bacteria doon. Because microorganisms love most of the microorganisms, example your bacteria, love wet environments because they are made mostly of water. And once they are dehydrated, mamamatay sila. Okay? So, this, ayan on the inside, the microorganism is like a tiny machine that needs water to run. So, parang water yung fuel ng microorganisms ninyo. Okay? So, you have encapsulated bacteria, usually, tama ba itong, ayan, encapsulated bacteria usually produces a smooth, mucoid, and glistening colonies of nutrient agar. Ito siya class, since encapsulated yung bacteria mo, meaning hindi siya dehydrated. It is very healthy. That's why the colony that it produces in a culture, okay, is smooth, sticky siya or mucoidal, and glistening. Very healthy. While, on the other hand, if wala siyang capsule, non-encapsulated bacteria siya, ang napoproduce niya na colony is dry and rough, unhealthy type of colony. Okay, so ito yung example natin here at below. You will see this is an example of an encapsulated bacteria. May capsule siya, protected by a capsule. On the other hand, sa taas niya, this is the example of a non-encapsulated bacteria. Okay, yan yung itsura nila class under the microscope. Okay, so see the difference? Napakalaki nung capsule nito compared to dito na wala. So this one will produce a healthy, smooth colony, while this one will produce a dry and rough colony. So we have an example here. Ito yung example ng smooth colony. Diba? Napaka-healthy. On the other hand, this is an example of a dry colony. Dead siya tingnan. Okay? So remember that. Now let's go to the second part, which is the second external part of the bacteria. You have the flagella. Okay, class. Ayan. Let's proceed to the flagella. Okay, when you say flagella, this means whip. Okay, latigo. Okay, so this is characterized as a long filamentous thread-like protein appendages used by bacteria for movement. So the main function of your flagella is for locomotion. Okay, movement of your microorganisms. So they are made up of proteins class. Okay. One example of the protein or your flagella consists of a protein called flagellin. So, ano ba yung flagellin? When you say flagellin, globular protein siya, okay? The shape is globular. Na take up nito sa biochemistry, it is a globular protein, okay, that is arranges itself in a hollow cylinder to form the filament in a bacterial flagellum. Now, just remember when you say flagellin, globular, it is a globular protein that comprises the filament of your flagella. Okay? Arise from a basal body in the cytoplasm, meaning ang flagella ninyo, saan siya nag-originate? Sa cytoplasm of your prokaryotic cell. It originates sa cytoplasm and projects outwardly through the plasma membrane. So, it passes through your plasma membrane, followed by the cell wall, and finally the capsule. Okay? Attached to protoplasmic material inside the cell. So, when you say protoplasm, okay, mamaya na, I will just explain the difference of the protoplasm, okay, from your cytoplasm and so on. But mainly, ang sinasabi dito, your flagella, it arises from the cytoplasm and passes through the three coverings of your microorganism. So, galing sa cytoplasm, pinapass through niya ang plasma membrane, followed by cell wall, and lastly, sa capsule. Okay? So, not all bacteria have the flagella, okay? The same with your capsule, hindi lahat ng bacteria natin merong flagella for locomotion. Okay, so however, for those that do have your flagella, the number and arrangement of flagella is characteristic to the species. So, depende sa species ng isang bacteria, ang number ng kanyang flagella will depend sa type ng species ng bacteria na yon. For example, the presence of flagella is used for classification purposes. So, nagagamit yung, you can use the appearance, the appearance, the location, and the number of your flagella as a classification to classify what? The type of species of the bacteria. So, magagamit mo siya as palatandaan. Okay, so you have 4 types of arrangement of your flagella. Number 1, you have the monotrichous. So, when you say monotrichous, one flagellum lang found at one end of the cell. Okay? Nakikita siya at one side and isa lang yung flagella. Now, when you say amphitrichous, single flagellum pa rin, nag-iisa, but at each end of the cell. So, left and right, merong isang flagella. Okay? Letter C, you have Lophotrichus. 2 or more flagella na ito, but are found at one end lang, or both ends of the cell. So, I will give you an example of the appearance later. And then lastly, you have the Peritrichus, flagella distributed all over the entire cell. Okay, so ito yung sample nila. Ito class, this is a picture. Pardon the bright, parang yung puti dyan sa gitna because I used a flash in. Take a picture of this from the book. Yeah. So, if you want, by the way class, if you want to find a study material, you can always find the book made by Tortora. Okay, Microbiology, Introduction to Microbiology. Ang kanyang author is Tortora, si Case, C-A-S-E, and nakalimutan ko yung isa pang, basta 3 sila class. Okay, so maganda yung book nila. Because related siya, I tried to use the book or as a material or reference material na pinakamalapit sa course pack natin. Okay, so you have this. Monotrichous, isa lang at one end. Amphitrichus, 1 per one end. Okay, left and right. And then you have the Lophotrichus, more than 1 or more than 2. Okay, pwedeng makita on both sides or sa isang side lang. And then you have the Peritrichus. Okay, the flagella is found all over the surface of bacteria. Ayan, so, microorganisms which have flagella, example natin, you have the protozoa, pwede ring certain algae, You also have certain species of bacilli, and species of Vibrio and Spirillum. Okay? So when you say Vibrio class, ito siya yung type ng bacteria. Ang shape niya is parang curve rod or comma. Parang comma yung shape niya. C-O-M-M-A. Okay? Para siyang comma. And these are found, your Vibrio, or the type of bacteria that are found in undercooked seafood. For example, sa sushi. Okay, ano pa ba yung sa Filipino? Yung nilalagyan lang ng suka? Isa yun. So, undercooked seafood, it may contain Vibrio. Okay? So, gram-negative bacteria yan sila. On the other hand, you have also your Spirillum. When you say Spirillum class, its shape is spiral. For example, ito. This one is a Spirillum. Spiral or corkscrew-like appearance or corkscrew appearance. Okay, spiral. So, gram-negative bacteria din yan siya. So, these two contains flagella. Okay, your cocci, these are the type of organisms or microorganisms that does not contain flagella. Okay, wala siyang flagella. Why? What is the shape of your cocci? Your cocci, this is a type of bacterium or archaea siya that has a ovoid or spherical round shape, bilog. So wala siyang you will not find flagella in round shaped bacteria. Okay? So yan. Now let's go to eggshell filaments. Okay, your eggshell filaments class, these are bundles of flagella-like fibrils. Okay? Para din silang flagella. When you say fibril, ano siya? Characterized as slender fiber. Thin, small, slender fiber. Okay? So, flagella-like fibril. Yung inyong eggshell filaments. Similar ang appearance niya sa flagella. So, it arises at the end of the cell beneath the outer sheath and wrap around the length of the cell. Now, there are certain types of bacteria class. You call them as sheathed bacteria. Okay? Ito na sheath ha. Yan. Sheathed bacteria. Covered sila with gelatinous material or gelatinous sheath. Now, under the sheath class, sa ilalim ng gelatinous sheath na yun, doon yung makikita yung eggshell filaments. Okay? Wrap around the length of the cell. Okay, so these eggshell filaments rotate like flagella. Ang kanilang movement class is rotation. Okay, the same with your flagella. But since they are under or they are found under the outer sheath, okay, na 'di ba napalibutan sila ng gelatinous sheath, this causes the outer sheath to and gives the bacteria a corkscrew motion. So remember, di ba? Your flagella is used for locomotion. That is also the function of your eggshell filaments. Pero since magkaiba sila ng flagella, because your flagella, di ba, outward, your eggshell filaments nakapalibot sila sa cell mismo. Now when they try to move, this gives your bacteria a corkscrew motion or spiral motion, kumbaga. So, saan sila nakikita itong eggshell filaments? They are unique to sporochetes. Okay, sporochetes. Now, your sporochete, these are actually, okay, nakikita lang yung eggshell filaments sa sporochete lang. Okay. When you say sporochetes, these are group of bacteria that have a unique structure and mobility or motility, I mean. One of the best known sporochete is your Treponema pallidum, okay? The causative agent of syphilis. Now, ano yung syphilis? I think this is very narinig nyo na ito. This is a very common disease. This is actually a sexually transmitted disease. Okay, so you can get syphilis through sexual contact. Okay, a bacterial infection that is spread by sexual contact. Now, your syphilis class is characterized by the presence of sore. Okay? Sore. When you say sore, yung parang medyo sore na part ng balat. Okay? So you can find the sores maybe on parts like your genitals, okay, the genitals, pwede rin sa may bandang rectum and sa mouth, around the mouth. Okay, so if bago nyo i-kiss yung mga partner ninyo, like your boyfriend and girlfriend, please check if meron silang sores found around their mouth. Okay, sore na S-O-R-E-S. Pag meron yan, baka mamaya merong syphilis yung boyfriend or girlfriend. Okay, so please check that out. Spread, okay. Your syphilis actually spreads from person to person via skin or mucous membrane contact with the source. So, hindi kailangan ng merong sexual penetration. Matouch mo lang yung part, okay, ng sore. Matouch mo yung skin niya or yung kanyang, the mucous membranes found on the sore. Pwede ka nang magkaroon ng syphilis. Okay, so be very careful. Have awareness, okay? Now, eukaryotic cells, they do not contain your eggshell filaments. So, walang eggshell filaments ang eukaryotic cell. Okay, this is the picture of your Treponema pallidum. Ito class, this is your eggshell filament. Diba? Nakapalibot sila sa cell mismo ng bacteria. Okay? Next, let's move on to the pili and the fimbriae. What's the difference between your flagella, your eggshell filament, and your pili or fimbriae? Okay, na ang biggest difference nila class, mas maliliit yung pili and fimbriae ninyo. Okay, so when you say pili or fimbriae, these are hair-like appendages attached to the bacterial cells in much the same way as your flagella. So meaning, saan sila na-originate? The same din from the cytoplasm and then passes through the plasma membrane, the cell wall, and the capsule. But unlike your flagella, they are considerably shorter and thinner and smaller. Maliliit, maninipis. They have a rigid structure. So, one big difference, your flagella class, diba? They are flexible. Okay, malambot. On the other hand, your pili and fimbriae are very rigid, matitigas, solid. Okay? They are not associated also with motility because their main function is for attachment. Okay, when I say attachment, your pili are used by your bacteria to attach to surfaces. Okay, so ano yung protein consisting your pili? It is called pilin. Okay, so fibrous protein itong pilin natin that are found in your pili or your pilus. Okay, next. Okay, so your pili and fimbriae are found on some negative or gram-negative bacteria. Now, kanina pa nababanggit itong gram-negative bacteria. Ano pala ito, ma'am? What is a gram-negative bacteria? Your gram-negative bacteria class, they're frequently nakikita sila sa enteric or intestines, okay? Sa loob ng intestines, yan sila. And your gram-negative bacteria, when you stain them, they will give a pink stain lang. Ang mabibigay ng gram-negative bacteria na color is pink color after the staining. Why? Because your gram-negative bacteria do not contain a thick cell wall. Okay? Kapag sinabi mong gram-negative bacteria, they will give a pink color to gram-negative staining because they do not have a thick cell wall. Now, what's the function of the thick cell wall? Okay? Yung thick cell wall kasi class, siya yung mag-absorb, okay? absorb the color of the violet stain from your gram staining method. Okay? So kapag may thick, may makapal na cell wall yung isang microorganism, it will absorb the violet stain, okay, from the gram staining method. Now, if the cell wall is thin, manipis lang, hindi niya masyadong na-absorb yung violet color. That's why the resulting color is pink color. Okay, so kapag sinabi mong gram-negative, manipis ang cell wall and it will just form a pink coloration after the gram staining method. On the other hand, when you say positive or gram-positive bacteria, these are the bacteria containing a thick cell wall. Sila yung makakapal ang cell wall. That's why when they undergo gram staining method, okay, they are able to absorb the purple or violet color of the chemical dye. Okay, so kapag gram-positive bacteria, ang color niya is purple or violet, meaning may makapal na cell wall. Okay, so please remember the difference between your gram-negative bacteria and the gram-positive bacteria. Okay, so eukaryotic cells, they do not have the pili. And they arise from the cytoplasm, extends, okay, nasabi ko na yan. Let's proceed. So this is the appearance of your fimbriae and your pili. Tandaan ninyo class, mas maliliit ang fimbriae at mas marami sila. Okay, on the other hand, yung pili ninyo iilan lang, 1 or 2 and mas mahaba. Now, let's go to the different functions of your pili and fimbriae. Number 1, they are used for attachment to other bacteria or to other membrane surfaces such as the intestinal lining. So, yan, main function nila for attachment. Number 2, they absorb additional oxygen and nutrients when the supply is low. So, ginagamit rin sila for absorption of nutrients. Number 3, site for the attachment of bacterial viruses. What is a bacterial virus? Okay, we call this type of when you say bacterial viruses, virus ito sila class, okay? They are the type of virus that inject their genome. Ano nga ang genome? Okay, the overall genetic material found in a microorganism. Okay, what happens in your bacterial viruses is that they inject their DNA or their genome into a host bacteria. Okay, nililipat nila yung genome nila sa isang bacteria, initiating what? The production of new viruses and viral DNA. So, ginagamit itong mga bacterial viruses natin, ginagamit nila yung bacteria as a host para paramihin yung kanilang DNA material and gumawa ng mga bagong viruses. Okay? So, you call your bacterial viruses as bacteriophage. Okay, bacteriophage, bacteriophage. When you say bacteriophage, this is a virus that infects and replicates within a bacteria or archaea. Okay, virus siya na nag-i-infect ng isang prokaryote like bacteria and archaea, and it replicates within the bacteria. Okay, so bacterial viruses. So, your bacterial viruses may connect, okay, it injects its genetic material to a bacteria using your pili. Okay, sa pili, ginagamit ng bacteriophage ang pili to inject its genetic material to the bacteria, because your pili is also called as the sex pili or the material for conjugation. Okay? Number 4, join bacterial cells prior to the transfer of DNA from one cell to another. Now, pwede kasi class, there are instances that your bacteria, they exchange genetic material. And this is what you call the conjugation process. Okay? Conjugation, transfer of DNA from one bacteria to another. They are able to do that through using your fimbriae and pili. Okay? Or mainly your pili. Number 5. Okay, your fimbriae. Okay, so ito na nga, example natin. We have the fimbriae and the pili. Okay, your fimbriae vary from yung kanilang number from a few several hundred. Okay, so napakarami nila. They are very much plenty outside the surface of your bacteria. They are used for attachment to surfaces such as the mucous membranes of your intestines. One example natin dyan is your gonorrhea or your Neisseria gonorrhoeae, okay? Neisseria gonorrhoeae, and your Escherichia coli or your E. coli. So these microorganisms use your fimbriae as a hanging on object to establish urinary tract infection. Now, take note of this class The main difference between your fimbriae and your pili is that fimbriae is used for attachment to surfaces and not to other living microorganisms. Ang pilay natin, ginagamit siya for what? For attachment to other bacteria and organisms. Okay? So, yan yung pinagkaiba nila. Surfaces ang sa fimbriae and then other bacterial cells ang sa pili. Okay? For attachment. Okay, your pili is a longer version. Okay? So ang present na pili sa isang bacterial cell is mainly 1 or 2 per cell. Okay? They are used to join bacterial cells in preparation for the process of conjugation or the transfer of DNA. Now, they are also sometimes called as sex pili. Okay? Okay, class, I hope I'm back. Nag-water break si ma'am. Okay, so, we're recording. Let's continue. Okay, the next topic in this is the cell wall. Okay, we're trying to know what the cell wall or what composes the cell wall of a certain prokaryote. Okay, so when you say cell wall, it is a complex semi-rigid structure that gives shape and protection to the cell. Okay? Mainly protection siya. So, complex. Complex yung ating cell wall. Why? Because it is composed of your peptidoglycan. Okay? So, you have different functions for the cell wall. Number 1, it protects bacterial cells from lysis in dilute solutions. So, it prevents cell from rupturing due to entrance of too much water. Now, when you say lysis, class, ito yung rupture of the cell wall, okay? Pumuputok yung cell wall ng isang prokaryote. Why? When your prokaryotes are in dilute solutions, class. By the way, what is a dilute solution? Ito yung solution that contains what? More solvent than dissolute. Okay? When you say dilute solution, it has a small amount of solute dissolved in a solution. So, meaning, mas maraming water kaysa sa iyong solute. So, when your bacterial cells are in a dilute solution containing more water, the water in the solution will enter, okay, your bacterial cell. Why? Kasi maliit lang yung concentration or mas maliit yung concentration ng water inside the bacterial cell compared doon sa concentration ng water sa solution mo. So, mayayari, mapupuno ng tubig yung loob ng bacterial cell. And if wala siyang cell wall, okay, there is no material that will protect it from the entrance of water in dilute solutions. So, kaya puputok siya, okay? Now, if meron siyang cell wall, hindi ganun kabilis na papasok yung tubig. So, walang lysis na mangyayari. Okay? So, this is following the process called diffusion, okay? 'Di ba? Sa diffusion natin, from a higher concentration, pupunta siya sa lower concentration. So, the water of higher concentration sa isang solution will enter the bacterial cell with the lower concentration of water. So, yun yung diffusion. And diffusion of water will be prevented if your bacterial cells have a cell wall. Okay? So, yan yung one function ng inyong cell wall. Second one, it helps give shape. Bakit? Kasi nga, semi-rigid yung structure ng ating cell wall. So, it helps give shape to the bacterial cell. It is also the point of anchorage for the flagella. It may contribute to the ability to cause disease. Why? Bakit? Because it protects, the main function of your cell wall is to protect your bacteria. If meron siyang cell wall, mas mahirap patayin, okay, yung isang bacteria. Okay, next we have the site of action for some antibiotics. So, yung cell wall ng isang bacteria class ang inaatake ng antibiotics. The target of the antibiotic is to destroy the cell wall of the bacteria in order for it to die easily. Okay, by engulfation. Okay, next. Site of action for some antibiotics. Okay, tapos na. Chemical composition may be used in classification. Correct. Why? When you say classification, meron kasi tayong two types class ng prokaryotes. Okay, we have the eubacteria. When you say eubacteria, meaning true bacteria. Okay, and then we also have the archaebacteria or your archaea. Now, your archaea class are the fake. Parang fake sila na bacteria. Why? Because, although pareho sila ng eubacteria ninyo na prokaryotic or unicellular, magkaiba ang composition nila when it comes to their cell wall. Okay? So, later on, matitake up natin yan. Now, when you say eubacteria, ito yung mga bacteria na true because they contain peptidoglycan in their cell wall. On the other hand, ang archaebacteria, they do not have peptidoglycan in their cell wall. So doon pa lang, the presence of the peptidoglycan in the cell wall will help you classify the microorganism, whether it is a eubacteria or an archaebacteria. Okay? Next. Structures internal to the cell wall. Okay, so these are the structures that are found inside the cell wall. Okay, so number one, you have the plasma membrane or what you call the cytoplasmic membrane. So this lies inside the cell wall and encloses your cytoplasm. Yun yung main function ng plasma membrane natin. Okay? It encloses the cytoplasm. This also serves as a selective barrier to substances entering and leaving the cell. So I think hindi na natin ito i-discuss. Na-discuss yun na ito noon kahit sa physiology anatomy and physiology pa. Next, we have the cytoplasm itself or this definition here, class, is a prokaryotic definition. Why? There is a difference between the cytoplasm of a eukaryotic cell and a prokaryotic cell. Okay, magkaiba yan sila. So, in the cytoplasm class, in the eukaryotic cell, yung cells ng animals and plants, 'di ba? Meron siyang hati. Okay, kasi meron pang nucleus, 'di ba? May nuclear membrane pa. Outside the nuclear membrane, you will find the cytoplasm. But in a prokaryotic cell, wala man tayong nucleus. So, the whole inside of the prokaryotic cell is just cytoplasm lang. Okay? Walang nucleus. Okay? So, meron tayong tinatawag na protoplasm. Okay? When you say protoplasm, this is the combination of your cytoplasm and nucleoplasm. Okay? Pag sinabi mong protoplasm, ito na yung the overall material fluid inside the cell. Now, the protoplasm, the protoplasm in your eukaryotes are just found outside the nucleus, while the protoplasm in your sandali, parang may something. Okay, cytoplasm in eukaryotes, protoplasm outside the nucleus. Nucleoplasm in eukaryotes. Okay. So, protoplasm, diba? The protoplasm is the overall material inside your cell. Okay? Now, example natin sa eukaryotes. Yung protoplasm found inside the nucleus, that is what you call, or found outside the nucleus, that is what you call cytoplasm. Okay, class? Baka nalito din ako, class. But, let's start from the beginning. Protoplasm, overall material, fluid material inside a cell. Okay? Now, the protoplasm outside the nucleus, you call that as cytoplasm. Okay? And then, the protoplasm naman found inside the nucleus, you call that as nucleoplasm. Okay? May two types tayo ng protoplasm in eukaryotes. Number 1 is the cytoplasm. Number 2 is the nucleoplasm. Now, anong pinagkaiba niya sa prokaryotes? Your prokaryotes does not contain nucleoplasm. Why? Wala nga siyang nucleus. So, sa prokaryotes natin, purely cytoplasm lang siya. Okay? So, I hope naintindihan. Medyo nalito rin si ma'am doon. So, sorry. Okay? Next, we have, there's no nuclear membrane for it to be outside of. So, yan na yun. Your cytoplasm in a prokaryotic cell is about 80% water and is thick, semi-transparent, and gel-like. So, yan yung kanyang composition. It contains dissolved and suspended materials such as your protein, your carbohydrates, your lipids, and inorganic ions. And it is also the site for any chemical reaction. Okay, class? So, yan yung sa ating cytoplasm. Next, we have the nucleoid. The nucleoid is the region of DNA concentration. Ito siya. Okay? So, as you can see here, di ba, ang cytoplasm niya walang hati. Wala siyang nucleoplasm. Cytoplasm lang lahat within the cell. And then we have the nucleoid here at the center. Okay, DNA concentration siya. Ito yung nagko-control ng heredity. Okay, so, yan. Next, let's go to, we have the ribosomes. So, ribosomes, this is a site of protein synthesis. I think alam nyo na rin naman ito. The same with your eukaryotic cells. Synthesis of proteins pa rin yung main function niya in prokaryotic cells. And the ribosomes are mostly scattered all over the cytoplasm. Okay? So, ayan o. Ribosomes, nakakalat lang siya sa cytoplasm. Next, we have the inclusions, or you also call this class as inclusion bodies. Okay? So, your inclusions, these are reserved deposits of chemical within your cytoplasm. Okay? Dito nagde-deposit ng extra chemical materials, okay? So, your inclusions tend to accumulate when nutrients are plentiful. If sobra-sobra yung nakuha na nutrients or naiimbak na nutrients ng inyong prokaryotic cells, they will store that in a form of an inclusion body. Okay? And then lastly, you have your endospores. So this is the appearance of a bacterial endospore, class. Ayan yung appearance niya. So when you say endospore, ito yung dati nating active na mga cells that experience lack of food source, for example, kaya naging dormant sila. Okay? So kaya the endospore, class, is the key to the survival of your bacteria. You might wonder, bakit napakatagal, ancient na masyado ang ating bacterial cells pero hanggang ngayon buhay pa rin sila. Hindi sila na-extinct. Why? Because of the use of the endospores. Okay? So your endospores are defined as dormant cells formed when certain genera of your bacteria, mainly your Clostridium and Bacillus, find themselves facing adverse conditions such as lack of nutrients, drying, presence of toxic materials, and so on. Kapag nakaka-experience na ng hardship, ng challenges yung inyong bacterial cells, they will undergo dormancy. Okay, cellular dormancy. When you say cellular dormancy, this refers to cells being inactive. Okay? Hindi naman sila namamatay, but they are not active. Okay? Para silang in a deep sleep. Okay? Kapag nagkaroon ng shortage of food, wala ng nutrients, nagkaroon ng drying, wala silang water na makukuha, and merong mga materials that might cause harm to them, they will undergo dormancy in order to survive. Now, once bacterial cells will go to dormancy and form endospores, it can withstand extreme conditions and they survive for apparently unlimited periods of time. Okay? Kaya sila nakaka-survive because they are able to become dormant. Parang ayoko nang i-face ang challenges ko, matutulog na lang ako. So sana ganun din yung humans, no, class, na whenever we are finding ourselves in a hot spot or hard situation, we just become dormant. So gigising ka lang if okay na yung situation. So ganun yung ginagawa ng bacteria natin. Okay? So, eukaryotic cells. So again, take up na lang muna natin to for the first part. Your eukaryotic cell structure and function. Mahaba na siguro masyado yung video. Sige, let's just take this on the second part na lang, class. We will start, para sa second part ng video, we will start in the eukaryotic cell structure and function. Okay? For now, that's all for the first part. I hope na you will take note a lot of the additional information I gave you, and please watch the second video, class. Okay, so that's all and goodbye.